This is Professor Doherty, and today I'll be covering the digestive system. So for the digestive system today, I will first start with a very brief overview. Then I'll move into a few different organs, the stomach, the liver, and the pancreas, and what they secrete and how they contribute to the digestive system. So first, an overview. Primarily, the way that the digestive system breaks down fats, carbohydrates, and lipids is through secretions through what are called exocrine glands. Now, the exocrine glands are going to secrete digestive enzymes, and those in turn are going to break down all the constituents that turn into nutrients for us. In addition to that, many of these organs can also secrete hormones as well, meaning that they contribute to what's called the endocrine process. So these are signaling chemicals. Typically, I'm not going to cover a ton of these, but I will have to cover some because some of the signaling takes place between different parts of the digestive system, whereas some of the signaling takes place between the digestive system and the brain. So let's start with how digestion works. So this is just a very brief overview of how digestion works. If we look here, we have a carbohydrate, and the way that the digestive system is going to break this carbohydrate down into simpler forms. So for instance, here we have maltose, and we want to break this into glucose, is through a process called hydrolysis. Hydrolysis, like the name implies, lysis meaning to break apart, hydro meaning with water, so to break apart with water. And the way that your body accomplishes this is by using an enzyme. So here we have an enzyme, and this enzyme is going to grab a hold of maltose, and through using water, it's going to separate this maltose into two individual glucose molecules. Now you can see where this water winds up. It basically winds up here and here on these two glucose molecules as it is added where the separation takes place. Now, in fact, your body does this for all different types of nutrients. So, for instance, here we're looking at the biomolecule of carbohydrate. Here is the biomolecule of protein, and here is a biomolecule of lipid. Now, if we take a look, the process is pretty much the same for all three of these. Here we have a peptide bond, and this peptide bond is going to be broken using water and a protein digestive enzyme. So the thing that we're really changing here, besides the biomolecule that we're looking at, is the enzyme that's being used. So the enzymes are specific to breaking either carbohydrates, proteins, or lipids. And in fact, these enzymes can be very specific to actually places on the proteins, carbohydrates, or enzymes as well. So in this case, let's just take a look here. We can see that this water molecule that we're using in this process is again going to wind up between these two now individual amino acids. And then finally for fat, you can see that here we're taking three water molecules using a digestive enzyme to break this down into three fatty acids and one glycerol molecule. Now we're going to start with the stomach. I realize that some digestion does begin immediately when you take something into your mouth. There's amylase in your mouth, and that begins to break down carbohydrates, but not a ton of digestion is done there. Much of it is done in the stomach and then in the following small intestine and large intestine. So let's take a look here. This is just the anatomy of the stomach. Many of you are probably familiar with this if you've had anatomy. If not, that's not a huge deal. But basically you can see the esophagus here. So this is the esophagus has been cut off here, but the esophagus leads down into the stomach. There's a region here at the top called the cardia, which is so named simply because it's close to the heart. The fundus of the stomach is up here. The fundus is a generic term for the top of an organ. We also have fundus of the uterus, for instance. This top portion of the stomach, though, has a tendency to collect gases that are coming off from the digestive process. Now, those gases can sometimes be released up through the esophagus, but oftentimes they'll also make their way down into the small intestine over here. You can see that the stomach, as we move down into the cross sections of the stomach here, there are actually three layers of muscles surrounding the stomach, and these help with moving the food and digestive fluids around during the process of digestion. Now this down here, we've had the top portion of the stomach cut away, so here we're actually looking into the stomach. You can see that there are gastric rugae here. These are basically these folds that are in between these kind of pillar parts here in the stomach. And down within these gastric rugae, we will also have what are called gastric pits. And those gastric pits are where our digestive enzymes come from. Now, one thing that you should understand is that the stomach has a lining of mucus. And it has a mucosal lining here as well, which is just part of the layering of the stomach. But this mucus, and you can kind of see it here in white, helps to protect the stomach cells themselves. And we'll talk about that more in just a little bit. Down at the bottom part here, we have a region called the pylorus, which comes just before the pyloric sphincter. The pyloric sphincter is what controls the flow of food from the stomach down into the small intestine. Now, the portion of the small intestine that the stomach attaches to is called the duodenum, and there are two other parts to the small intestine as well, which we'll talk about in a little while here. Gastric glands. So here what we have is a cross section of a portion of the tissue of the stomach wall. And so you can see the top portion of the stomach wall is made up of cells that are called the mucosal region. In this region, we have these pits shown here and also shown in a larger cross section over here. These are called the gastric pits. The gastric pits are important because they house the cells that actually make the digestive enzymes, and we'll go through these in just a moment. You can see that in this particular slide, we're showing three of these cells: mucosal cells, parietal cells, and chief cells. Now, down below the mucosal layer, we have submucosal layers consisting of some muscular strands as well as some connective tissue down here. And let's talk about some of these cells that are in the stomach. All right, so there's a number of cells involved in not only protecting the stomach but also creating enzymes. So let's go ahead and start with mucus cells. Like the name implies, mucus cells produce mucus. Now, mucus consists of glycoproteins and water. And this serves as a protective layer. So this protects. And it protects the stomach cells from hydrochloric acid that is produced by some of the other cells within the stomach. So this mucosal layer basically acts as a protective blanket covering the cells, protecting them from this acidic hydrochloric acid that's being produced for digestion. Now, if you recall, we did talk about how mucus production goes down when you block COX-1 enzymes. So COX-1 enzymes are actually in the production line for making mucus. So if you take aspirin, you can actually reduce your mucal output in your stomach, reducing the protection on your stomach walls. Second type of cell are called parietal cells. Now, parietal cells produce hydrochloric acid, like we just talked about. And they do this in response to histamine. So basically, when they get hit with histamine, the parietal cells begin to produce hydrochloric acid. This contributes to the degradation of the food that's been eaten, so it helps to break up the food. Hydrochloric acid is also used as a trigger to help trigger certain sorts of protein digestive enzymes as well. In addition to this, parietal cells will also produce what's called intrinsic factor. And this intrinsic factor is necessary for B12 absorption. So that's vitamin B12. Vitamin B12 plays a variety of roles. It's key to normal brain function and nervous system functioning, and it's also involved in metabolism. You can get vitamin B12 from fish and liver, so like meat, poultry, eggs, milk, and some bacteria. So you can see how some of these cells are not only contributing to the breakdown of biomolecules, but they're also helping with absorption of other things that we need, vital nutrients. The third type of cell is called a chief cell. Now, chief cells make pepsinogen. Now, you might recognize ogen. So ogen means that, so this is OGN here. If you recall, when a word has OGN at the end, that means that it is an inactive enzyme. So this is an inactive precursor to the enzyme that is called pepsin. Now, pepsinogen can get converted into pepsin when it comes into contact with hydrochloric acid. So you can see here how the hydrochloric acid from the parietal cells is going to trigger the production of pepsin. Now, pepsin is an enzyme that digests proteins. Okay, so pepsin digests proteins. So basically, it cleaves proteins, helping to cut them down into smaller parts. Some other cells that also contribute to digestion, let me move this up a little bit here, include ECL cells. And this stands for enterochromaffin like cells. So basically ECL. So enterochromaffin like cells. These cells are within the lining of the epithelium of the stomach, the small intestine, and the colon. So you can find these actually in stomach, small intestine, and colon. So these cells are widely dispersed. What they do is they produce serotonin. And serotonin, if you remember, it can actually go to the brain and be used by the pineal gland. But typically the serotonin that's produced here will also be used for cell signaling. So telling cells to do certain things like turn off or on, for instance. The cells down here, the ECL cells in the digestive system, produce about 90% of the serotonin in the body. The other thing these ECL cells do is they produce and release histamine. Now, this production of histamine is in response to another signaling chemical called gastrin, or hormone, I should say, called gastrin. And in this case, the histamine is used to signal parietal cells. And when I say it's used to signal parietal cells, what I mean is that it's used to signal parietal cells to begin production of hydrochloric acid. So you can begin to see how many of these cells are kind of related to each other and their functioning. It kind of ties in with each other. Next type of cell, this is our fifth cell, is called a G cell. G cells produce what's called gastrin, which is that hormone. Hence the name G cells. So, as I mentioned, gastrin is a signaling molecule that goes into the blood even, so it's called a hormone. And this goes out to stimulate the release of histamine from the ECL cells. Now, gastrin is typically one of the first hormones that's released during digestion, and gastrin can be released, or it can be stimulated to be released by either proteins in the stomach or by acetylcholine. And when I say acetylcholine, I mean acetylcholine from the vagus nerve. So this is a parasympathetic type of innervation or excitation. And remember, parasympathetic is our rest and digest system, so it kind of makes sense here that the parasympathetic nerve would stimulate these G cells to go ahead and begin production of gastrin, which kind of kicks off a lot of this digestive system starting up. Okay, last type of cell here are called D cells. And D cells stands for delta cells. And these delta cells produce what's called somatostatin. And they do this in response to gastrin. So gastrin release causes these cells to release somatostatin. Now, somatostatin is going to act on parietal cells. So somatostatin actually acts as almost an off switch. So it turns a number of things off. So it's going to act on parietal cells and it's going to decrease hydrochloric acid production. It's also going to, on like G cells, it's going to decrease gastrin production. As well as it's going to decrease histamine production. So you can see here that, in fact, somatostatins are actually almost acting like a molecular down rev, so to turn off the digestive system, or to decrease its functioning at least. All right, so now I know that was a long list, and so let me kind of give you a layout of this that's a little more simplified. So let's go ahead and cut this off here. So you can imagine that in the beginning, usually what triggers this digestive process are G cells. So we have G cells. They're being triggered by proteins or acetylcholine can trigger this, right? The G cells are going to produce gastrin, and gastrin in turn, so I'll put that here, so it's producing gastrin, and gastrin is actually going to go on two different pathways here. And this gastrin, so we're going to start down below here. And so this is basically the digestive portion down here. Gastrin is going to turn on ECL cells. And these ECL cells are then going to produce histamine. And this histamine is then going to activate parietal cells. Now, this causes our parietal cells to make hydrochloric acid. So now we have hydrochloric acid, and this is going to combine with pepsinogen, which is coming from chief cells. So chief cells are producing this pepsinogen. Now the combination of these two is then going to result in pepsin. And pepsin is an enzyme. And the result of it is that it winds up cleaving proteins. So the result of all of this train here in the end is simply the cleaving of proteins in the stomach. So in other words, the digestion of proteins in the stomach. Notice that in the stomach, we are not spending a lot of time digesting carbohydrates or digesting lipids. There is a little bit of that going on due to hydrochloric acid and some of the amylase from the mouth getting down into the stomach. But really, the point I'm trying to make is that your stomach is primarily set up to digest proteins, in fact. All right, so now let's go back here to the beginning, and we'll follow the other pathway here, which is much shorter. The G cells, when they produce gastrin, will eventually have an effect on D cells. And these D cells are then going to produce, so they're going to make somatostatin. Now, if you remember, somatostatin acts as, it's a molecular off switch, right? And so now these somatostatins are going to go and act on a number of different cells. So they're basically going to go and turn the parietal cells off, as well as they will have a negative effect on the G cells, so they'll turn them off, right? So this is basically a negative feedback loop. I'm just saying off here though, right? As well as it will go and turn off the ECL cells. And even if it doesn't turn them off, it will reduce their functioning at least. So you can see here how the progression, this chain of progression from G cells turning on to eventually D cells turning everything off takes place within the digestive system. And this is due to either having food in the stomach, which turns things on, or the food in the stomach being gone, which eventually turns things off, or by parasympathetic activity or inactivity. So if we come back to this picture, you can see here where some of these cells are located. So when we talked about the mucous cells, they are at the top of the gastric pits, which makes sense because you want the mucus to be on the top layer here protecting all of these cells. And then down in these pits, we have parietal cells and chief cells. So they're producing things like hydrochloric acid, very caustic type of enzymes and chemicals that will eventually work their way up the pits and on top of the mucal membrane, so that staying on top of the mucus, hopefully, so they don't damage the cells below them. Now, another thing about this mucus up here that I didn't mention before is that it is somewhat alkaline. So it will offset the acidity of the parietal cells, coming off the parietal cells. And so this alkaline that's in the mucus, it does not immediately offset the acidity of the stomach, so the acid can come out and interact with the biomolecules like proteins and carbohydrates. But if the acid turns around and comes back towards these cells, because they have this alkaline protective layer, it will offset that, neutralize essentially that acidity, and cause less damage to these cells on top. As you might imagine, these cells turn over quite frequently just due to the damage from all the caustic chemicals and enzymes that are up here. All right, let's take a look here at how bicarbonate is actually created through this process of creating acid. So this is actually kind of interesting here. If you look here, this is a picture of a parietal cell. And remember, the parietal cells need to create acid. And the way that they do this is through the bicarbonate equation. They take carbon dioxide and water, and using carbonic anhydrase as the enzyme here, they make carbonic acid. And carbonic acid results in bicarbonate and, of course, hydrogen ions, which is our acid. So let's start with this. The acid will then make its way to the apical surface of this cell. Now remember, up here is the inside of the stomach, so where we want digestion to take place. The cell will then pump hydrogen out of the cell in exchange for potassium from whatever is in the food up here. This hydrogen will move out of the cell in exchange for potassium. Now that we have hydrogen in the lumen of the stomach, chloride will follow it, and it will just follow it due to diffusion. Now on the other side down here, on the basilar side of the cell, we're now getting a buildup of bicarbonate. And because bicarbonate is useful to us as a buffer in the blood, what these cells will do is they'll actually exchange bicarbonate out into the blood in exchange for chloride into the cells. So you can see that we almost kind of get two good things here. This is actually a pretty good process where we're getting more acidity in the stomach, and as we get more acidity in the stomach, we get more bicarbonate and more buffer in the blood. So it's kind of a win-win situation here. Now let's take a look at some of the functions of what we call gastric juice. So gastric juice is just a combination of all the enzymes and fluids that are produced in the stomach. A typical pH in the stomach is approximately 2. I've seen it listed as 1 before, and that's fine, a pH of 1, but note that this is very acidic on our pH scale. Remember that neutral is 7, 14 is very basic or alkaline, so we are very acidic here. If you actually go down right to the surface of the parietal cell where the acid is being made, the hydrochloric acid, you can even get as low as 0.2, and that's very, very acidic. What the acid does, remember, its primary purpose is to convert pepsinogen into pepsin. Remember, pepsinogen is our inactive enzyme, pepsin is our active enzyme, and this enzyme is going to denature or cleave proteins. Now, this acid does a number of different things as well, so don't just think that the only thing that it does is help to produce pepsin. This acid helps to kill any bacteria or viruses that might have accidentally made their way into the stomach. You think about all the stuff you eat, there's bound to be some bacteria and maybe some viruses on the things that you eat, maybe even parasites, and this acidity, this low acidity, helps to kill those bacteria and viruses. So here in the stomach is where we really start protein digestion. Notice that we don't have any enzymes for carbohydrates and nothing for lipids. So, some of the acidity of the stomach may begin to break down some of these carbohydrates and lipids, but there are no specific enzymes for those. Again, the acidity helps to protect us from pathogens. That's what I was just talking about. And then to protect us from our own hydrochloric acid, we have that mucus that I was just talking about. It's somewhat alkaline, and so because of that, it does contain some bicarbonate to help offset the acidity of the hydrochloric acid. In addition to that, the cells of the stomach use what are called tight junctions. So you may have seen this before in other classes where epithelial cells will bind very tightly to each other. And what this does is this prevents anything from slipping between the cells. So for instance, if I were to draw two epithelial cells here, if this area here is very tight, it makes it so that we don't get any acid or anything like that slipping down in here and destroying any underlying tissues. Now, as I mentioned before, these epithelial cells also rapidly divide, and so because of that, they can replace themselves as they become damaged. Now with that, there can still be problems in the digestive system. Here what we're looking at is a reflux disease. In this case, this one that we're looking at here is called GERD. That means gastroesophageal reflux disease. And this is when acid gets past the gastroesophageal sphincter and up into the esophagus, and it begins to damage the lining of the esophagus. So if you look at this picture here, what this is a picture of is someone has taken an endoscope, which is one of those long tubular scopes that has a camera on the end of it, and they've stuck it down someone's esophagus. And here at the end, you can see that this is the gastroesophageal sphincter. Beyond the sphincter is the stomach, but what we're looking at here is actually the lining of the esophagus. You can see how this lining has been eaten away, it looks like. The reason for this is because this sphincter here is not closing down tight enough, and that's allowing acid to get back up into the esophagus, and this is what we call acid reflux. Another name for this that you may have heard of is heartburn. That's typically a lighter version of this problem. And typical treatments for this include antacids. So an antacid is basically something that's going to neutralize the acids in the stomach. You can also take what are called hydrogen receptor blockers and proton pump inhibitors, which basically stop the protons from being pumped into the stomach from the parietal cells. And anywhere between 10 to 20% of the population actually has this problem, so quite a high number of people. A more severe version of this is called Barrett's esophagus. And this is where you actually get modification of the cells here of the esophageal lining. And about 5% of people who develop this are likely to develop cancer. And basically what happens is over time, as acid reflux continues into this area, it irritates these cells, and these cells will actually change the type of epithelial cells that they are. And when they do this, they can turn into what's called an adenocarcinoma, which is basically a cancer of the epithelial tissue here. And of course, that is deleterious. And so if we look at some of the treatments for this, some of these I already talked about. So a number of things that you can do to treat these type of symptoms. And remember, 10 to 20% of the population has this, so they're probably undergoing one of these types of treatments. The most common is over-the-counter antacids, which just basically neutralize the acid in the stomach. However, you can also take what are called proton pump inhibitors, which, like I mentioned, inhibit the proton pumps in the parietal cells so they don't push those hydrogen ions out into the lumen of the stomach. And then you can also take what are called H2 blockers, and these also reduce the amount of acid that's pumped into the stomach. Now, one of the interesting things that they've discovered, I think probably in the last 30 years or so, if I remember correctly, is that one of the biggest causes of ulcers is actually a bacterium. And we didn't know this for quite a while. We just thought that the reason that people got ulcerations in the stomach was because they had a weak lining or it was genetic or something like that. But in fact, what they found is that quite a lot of stomach ulcers are caused by this bacterium. This is a gram-negative bacterium. And in fact, this bacterium has been found in 50% of the world's population in their stomach. So half of the people in the world have this bacterium in their stomach, but about 80% of those people who have this are asymptomatic. So in other words, this bacterium doesn't do anything to their stomach lining. So for the 20% of those 50%, so that would be about 10% of the population that has this, and this bacteria does affect their stomach, what it does is it basically reduces the lining of their mucus, or the amount of mucal lining that they have inside of their stomach, and this allows acid to then be able to attack their own cells or degrade their own cells. And this, of course, is what we call an ulceration. So in fact, I have heard of people taking like antibiotics to try to knock down the numbers of these pylori bacterium. All right, so that is the stomach. Now I'm going to move down the digestive system into the small intestine and the colon. The small intestine is made up of three parts, and two of those parts do a very similar thing, and the third one does something slightly different, so I've broken them out in this way. The first two parts of the small intestine are the duodenum and the jejunum. Here, what happens is these parts of the small intestine are mostly for absorption of carbohydrates, absorption of amino acids, absorption of lipids, and then absorption of a few different ions. So you can see in this section, in this first section of the small intestine, which is quite long when you count the jejunum, we're mostly absorbing biomolecules. So essentially what we're getting, this is all calories, right? So we're absorbing calories mostly, as well as a few critical nutrients here. In the final part of the small intestine, which is the ileum, you can see that we change to absorbing a few different things. So here we reabsorb some of the bile salts that were used in the beginning section up here. We absorb vitamin B12, which you recall intrinsic factor is important for absorbing this vitamin. We absorb electrolytes. So remember, electrolytes includes things like sodium, potassium, pretty much anything that has a charge on it. So chloride, things like that. And then also we begin to absorb water as well. Next, the food moves down into the large intestine. And again, the large intestine is also going to be used for absorption. But here the large intestine is primarily used for absorbing water. That is the big thing that the large intestine does. It will absorb electrolytes, so sodium, potassium, chloride, many of the B complex vitamins, vitamin K, and folic acid. So in fact, and again, the largest thing that the large intestine absorbs is water. That's the big thing that it absorbs. Now the large intestine can actually be used to excrete water as well when it's irritated. So if you think about cases of diarrhea where your feces becomes very fluid filled, this is because the large intestine is actually pushing water into or pulling water into itself to clean out its feces. This is typically due to an infection, and this is a response by the colon or large intestine to try to clear out the bad bacteria that have invaded the large intestine. So diarrhea is really your body's way of trying to push out the bad bacteria. The downside to this is that if you lose enough water through diarrhea, you can actually die of dehydration. So this can be deadly. Now, death by dehydration by diarrhea in first world countries like ours is very unusual. But in third world countries, this is a real risk. Quite a lot of people actually died simply from having diarrhea. Now, the other interesting thing I want to point out here is vitamin K. So vitamin K cannot be produced by our own cells in our body, and so we have to absorb it. And one of the interesting things is that the large intestine, remember, is host to a huge number of bacterium. And those bacteria are actually good bacteria. They live with us. They're part of what we call our flora. And when we eat food, there's always some undigested food that makes it down into the large intestine. And those bacteria in the large intestine will digest that extra food. And one of the byproducts of those bacterium is vitamin K. And we will then absorb that vitamin K. So the thing I'm trying to get across to you here is that the bacteria that are in your large intestine are actually vital to your survival because they produce things that your body normally can't produce and that are very difficult for you to get through your own food. Now here you can also secrete water, like I was mentioning before. This is typically due to active transport of sodium chloride. So for instance, if we want water to move out in the feces, we can put more sodium chloride into the feces and that will attract water to it. Remember that water has a tendency to heavily chase after sodium. And then the other thing about the large intestine is because it is host to so many bacteria, we have to have a good immune system around that large intestine in order to keep those bacteria in check, because we don't want those bacteria to get into our blood supply, for instance. So around the large intestine, we have these lymph nodes that are termed MALT, and MALT just means mucosa-associated lymphoid tissue. They're just basically lymph nodes. These lymph nodes are full of lymphocytes, and these lymphocytes then look for any bacteria that are making their way into the body, and they try to kill those bacteria before they get into our blood supply, get free in our body. So there is a significant presence of the immune system around, in particular, the large intestine, but all around the digestive system. And that's basically to protect you from those bacteria actually getting into our blood. Now, the final type of protection that the large intestine has are goblet cells. And goblet cells produce mucus. So much like the mucal cells in the stomach, these goblet cells are in the large intestine. They produce this mucus. The mucus lining helps to protect the cells of the large intestine. And in fact, you can see this mucus if you ever look at your feces after you've gone to the bathroom. Sometimes you can actually see this mucus lining on the outside of your feces. In particular, you will see this if you've been ill, particularly with a digestive illness. You can oftentimes see that the mucus has become much more intense and much thicker oftentimes. All right, so the next organ that I want to talk about that contributes to the digestive system is the liver. Now, the liver is a very large organ in your body, sits on the right side of your body. And you can see it here. It actually has an interesting blood flow that I'm showing here. And so what I wanted you to see here is that here is the small intestine. So you have food traveling through the small intestine, so food that's being digested here. And contributing to that digestion of that food is the liver through what's called bile. So the liver will produce bile. The liver stores bile in the gallbladder. So you probably heard of this. This is a small pouch that typically normally would sit here underneath the liver. Here we pulled it out so that you can see it better. So the liver is going to produce bile, store it in the gallbladder, and then as food makes its way down the small intestine, it's going to squeeze bile out of the bile duct here and onto the food. Now the purpose of bile is to emulsify fats. So let me be clear about this. The bile does not digest the fats, it only emulsifies it. And what that means is that it breaks it up into smaller droplets so that it can be more easily digested later on. So again, bile is just breaking up or emulsifying the fat. It's very similar to dish soap. So if you've ever put dish soap onto, for instance, an oil, like olive oil, you can see that it breaks up the olive oil. Same sort of thing is happening here. So bile is then going to enter your food flow here in the small intestine. Some of this bile, what's being shown here, some of this bile will continue with the food and eventually make its way out of the body as part of the feces. But some of the bile gets reabsorbed through what's called the hepatic portal vein. Now this is a unique type of vein, so I'm going to describe this here real quick. The hepatic portal vein runs from the small intestine up to the liver, and its purpose is to draw nutrients off of the food from the small intestine and take them for processing to the liver. In this case, we're also seeing that it's reprocessing some of its own bile as well, which is a side effect of this. But notice here that this is a unique type of vein because it runs between two organs. It's running between the small intestine and the liver. Most veins run between the organ and back up to the heart. So for instance, we actually have what's called a hepatic vein, which is not shown on this picture here, but if I drew it as a pen, the hepatic vein would come off and go back up towards the heart. And I should have drawn that in blue, but whatever. So you can imagine that this is the hepatic vein. The blood flow is coming out of here and going towards the heart. Notice that this one, hepatic portal vein, is going between, again, between two organs. So it's an unusual blood flow. So we actually have blood coming into the liver from the hepatic artery. We have blood coming into the liver through the hepatic portal vein. And then we have one way for blood to go out, and that's through the hepatic vein, which I've drawn up here on top. All right. So in fact, there are two veins, one bringing blood in and one bringing blood out of the liver. Again, the reason why this one is here is so that we can get nutrients off of the small intestine and immediately get them processed in the liver. In addition to that, because we have this vein, sometimes bad things like bacteria or viruses might get through here, and so because of that, the liver does have some immune cells within it to help look for any invaders that might have made it into the blood here. All right, so let's talk about the bile production of the liver. Typically, the liver will produce anywhere between 250 and 1.5 liters of bile per day. So 250 mils and 1.5 liters per day. So make that an average of about 750 mils of bile production per day. So that's a pretty good amount. The pigment in bile is called bilirubin. And this bilirubin pigment actually comes from hemoglobin. So that's what this is here. The liver is used to break down old blood cells. And what it does there is it breaks down these old blood cells and it breaks out the heme group. The liver typically keeps the iron for itself, but it needs something to do with the heme group, and it turns it into bilirubin, which turns into being the pigment that's in bile. Now, once this bilirubin gets down into the intestinal tract, so the small intestine, there are bacteria in the small intestine, and they will actually digest this bilirubin into what's called urobilinogen. Now, this urobilinogen can then either continue out of the body with the feces, in which case it actually turns the feces brown. So the brown look of your feces actually comes from urobilinogen, which is from bilirubin, which is made from the old heme groups from your blood. So in fact, the brown in your feces is due to the breakdown of your own blood. Now, this urobilinogen can also get into your urine, and it turns your urine yellow. And the way it does this is some of this urobilinogen is going to be reabsorbed back through the hepatic portal vein, and so it's going to be in the blood. When the kidneys find urobilinogen in the blood, they filter it out, they put it in the urine, and that makes your urine yellow. Additionally, bile can get turned into what are called bile acids and bile salts. I'm not as concerned with these. Just understand that basically bile acids and salts are, forms of bile, essentially. So again, if we look at this on this general picture, we can see how urobilinogen is going to affect the color of both the feces and the urine here. So remember, bile is being made in the liver. With that is bilirubin that's being made out of old blood cells. The bacteria in the small intestine are going to make urobilinogen. Some of that's going to go out in the feces, making the feces look brown. Some of it will be reabsorbed through the hepatic portal vein, and some of it's going to stay in the blood supply, and it will be filtered out by the kidneys, and that makes your urine look yellow. So this basically just talks about what I was talking about on the prior slide. Let's talk about some of the other things that the liver does. Blood detoxification. So many of you are aware that the liver is used to detoxify the blood. Here it will take out things like steroids and drugs out of your system. Many of you are aware that the liver is used to take alcohol and degrade alcohol out of your blood. People who drink too much alcohol, this is why they wind up with liver disease, is because basically their liver just can't handle the amount of alcohol that they're drinking. Alcohol is in essence a toxin to your body. Now, in addition to blood detoxification, your liver will secrete bile. It also has Kupffer cells for phagocytosis, as I mentioned. So these are some of the white blood cells that look for anything that's coming in through the small intestine and getting up into the liver. So the liver is used to break down chemicals. Ammonia is produced by what's called deamination of amino acids. So this is just part of the breakdown of amino acids. And then that ammonia gets converted into urea. So in fact, it's a lot of the urea is actually either coming from amino acids or the breakdown of fats. So fats and protein breakdown essentially. And an in-between step of that is ammonia. The good thing about urea, the reason why your body converts ammonia to urea is because it's not as toxic and it's water soluble. Ammonia is more toxic to your body than urea is. Now, I've talked about other animals like birds. Birds actually excrete their ammonia as what's called uric acid. And that uric acid comes out as a solid, And you've seen it, as we showed in bird poop, where the bird poop looks white. This is due to uric acid. So they make uric acid, we make urea. Now some small mammals, like I mentioned before, that live in the desert can actually also make uric acid. And in those cases what they do is they push the uric acid out as a pellet. And so in that way they don't need as much water as we do. We need water in order to be able to put urea in it, in order to get rid of that urea. Here's a list of liver functions, and you can see here that the reason why I put this up is because the liver just does so much stuff, and this is only a part of what the liver does. So I just wanted to show you that the liver is involved in a whole lot of different functions. You should know the main functions over here. You don't need to know all the small side functions over here. So you should understand that the liver detoxifies the blood. It's involved in the metabolism of pretty much all the biomolecules and the secretion of bile. All right, so let's talk now about glucose, triglycerides, and ketones. So the liver deals with all three of these different types of biomolecules. So let's talk about what it does with these. First of all, here are some terms that you should know in regards to what the liver can do. So first of all, glycogenesis. Like the name implies, so genesis means to create, glyco meaning glycogen. So the liver can create glycogen from glucose. So that's what glycogenesis is. So creating glycogen, which is our long chain storage form of glucose, from glucose. Glycogenolysis. So glycogenolysis, or glycogenolysis, however you want to say it. Lysis meaning to break down, glycogen at the first part here. So this means that the liver can break down glycogen into glucose. So the liver can do either one of these two things. It can either take glucose and turn it into glycogen, or it can take glycogen and turn it into glucose. These are just the terms that cover that. It can also do these other things as well. Gluconeogenesis. I've mentioned this one before. This is the creation of glucose from non-carbohydrate substrates. So again, the creation of glucose from non-carbohydrate substrates. When I say non-carbohydrate substrates, what I mean are lipids and proteins. So what I mean by this is that you can take lipids and proteins and actually turn them or convert them into glucose. Now, this takes more energy, especially proteins into glucose. It takes more energy, so you don't get as much energy out of the glucose molecules overall, but you can do it. And then finally, your liver can also do lipogenesis. And this is just the creation of fatty acids. So in other words, your liver can create fats also. Free fatty acids. So here, the liver can take free fatty acids and convert them into what are called ketones. Now, why would your liver do this? It's because ketones can then be converted into acetyl-CoA. And if you remember acetyl-CoA, that can be used in the Krebs cycle. So basically you can take these ketones and eventually pump them into the Krebs cycle. Now ketones can also be acidic, so overproduction of ketones, converting free fatty acids into ketones too much, can actually create an acidic environment. And as I've mentioned before, people with diabetes sometimes have this problem of doing too much conversion of free fatty acids to ketones. In addition to this, another important thing that your liver does is it creates albumin. And if you remember, albumin makes up much of the plasma protein concentration. And this contributes to colloid osmotic pressure, which we talked about when we talked about the blood. And this is the way that your blood is able to draw back in fluid. So if you recall, as blood travels into the capillary beds of the organs, so some of the blood plasma gets pushed out and becomes extracellular fluid, we need to recapture that at the end of the capillary beds. And albumin is critical to that. And so albumin is made within the liver. The other thing that your liver does is it creates globulins, all of them except immunoglobulins. So it'll create globulins, but not antibodies and not anything that's used in the immune system. These globulins are typically used to transport steroid hormones or sterols. You remember how those types of molecules do not transport well on their own in blood, so they need some form of protein to stick to. Those are globulins. And then finally, the liver also produces fibrinogen. So notice again, this is ogen, so it's inactive. This is an inactive form of fibrin, and that, when it's activated, it contributes to blood clotting. So it works with platelets to form blood clots. So again, the reason why I did this list is because this list are the really important things that the liver does, but it also shows you just how diverse your liver is as far as its functions. It just does so much stuff. So finally here, I'd like to talk about the pancreas and some of the enzymes that it creates. So I'm not going to spend a lot of time on this. This is my last slide. I just want to talk about some of the important enzymes created by the pancreas. You can see here are a list of enzymes that are created by the pancreas. They do a number of things. Basically, most of them are used to cleave biomolecules in the process of digestion. There are really only a few here that I want you to know, because I think a lot of them really don't matter for you as you go out into healthcare. But you should know these two, so let me circle these. Lipase and amylase, because these are the big two for digesting fats in the case of lipase and digesting carbohydrates in the case of amylase. And then the only other one I really want you to know here is trypsin, because that is one of the enzymes that cleaves proteins. So you can see here it cleaves peptide bonds. That means that it cleaves proteins. You remember that we had pepsin, that was in the stomach, and then as soon as the chyme, which is basically the digestive food, leaves the stomach and goes into the duodenum, it's going to get hit with trypsin, which will cleave the proteins in a different place than what the pepsin cleaved it in. And with that, that is the end of my lecture.